Welcome everyone to today's webinar. My name is Marie Latulippe and I'm pleased to serve as the Director of Science Programs at the Institute for the Advancement of Food and Nutrition Sciences, or IAFNS. And this webinar is the second in the series that we have planned with the USDA's Agricultural Research Service, Beltsville Human Nutrition Research Center, to showcase the great work that is happening at this center that is of keen interest to the nutrition community. And for those of you that might want a refresher on IAFNS, we are a nonprofit organization that advances food and nutrition science for public benefit. And the way we do that is we bring together scientists from government, industry, and academia, and we identify areas of common interest and develop research projects around those interests. We fund actionable science. That's the focus of those research projects. And we aim to convene experts of varying perspectives. And the way that we activate our projects is that we leverage pooled industry funding and we combine that with in-kind contributions from both the public and the private sectors. So we use this method to catalyze science that matters. And we want to let everyone know that this webinar is approved for one continuing education credit by the Commission on Dietetic Registration, and we'll let you know how to access that certificate at the end of the webinar. Very quickly, logistics for this webinar. We are going to run through all of the presentations, and then as time allows, we'll take some of your questions. But as we go along, if you have a question that arises for a specific speaker, feel free to type that into the questions box on your control panel, and we'll revisit those at the end. So here we have Dr. Naomi Fukugawa, who has joined us. So welcome, Dr. Fukugawa. And she is the director of the USDA ARS Beltsville Human Nutrition Research Center. She also serves as an advisor to the IAFNS Sodium in Food and Health Implications Committee. So we're very pleased to have you, Dr. Fukugawa, and we'll hand it over to you to kick things off.
Great. Thank you very much, Marie. And we all appreciate the opportunity to highlight some of the research being done by BHNRC scientists. This next slide shows the advisory committees on which I serve. As Marie has mentioned, the Agricultural Research Service is the intramural research agency of the USDA, supporting research from the bench to the field to the plate and to the impact on health and back again. On this next slide, we can see that the BHNRC, or Beltsville Human Nutrition Research Center, is one of 6 human nutrition research centers in the USDA ARS. BHNRC is the oldest and largest, located in approximately 6,500 acres between Washington, D.C. and Baltimore. HNRCs conduct intramural research and supports collaborative research with academia and industry focused on food, its components, and impact on wellness of the population. There are 3 fully federal HNRCs, including Beltsville, Grand Forks, and one on the UC Davis campus in Davis, California. In addition, we have 3 cooperative human nutrition research centers associated with the Baylor College of Medicine, Tufts University, and the University of Arkansas. We hope on the next slide that you have registered or will consider joining us for the complete series of webinars highlighting BHNRC research. We've covered added sugars on October 26th, and that recording is available online. Today, we're covering complex carbohydrates, and then this will be followed by talks on botanicals and flavonoids. So on this next slide, we ask the question, why complex carbohydrates? The simple definition of complex carbohydrates are that they're sugar molecules strung together on long, complex chains, and this presents some analytical challenges. In addition, complex carbohydrates take longer to digest and are a more stable source of energy, and hence an important part of one's diet. We all know that all carbohydrates are reduced to glucose, which is then used as energy by the cells. I think it's very important to emphasize that there's no real quotes good or quotes bad carbohydrate, but one must consider the type of food containing the carbohydrate of interest. Some simple carbs occur in healthful foods such as milk and whole fruits, and complex carbs also appear in foods with low nutritional value. So we must consider the whole food rather than just the carbohydrate that it contains. So focusing on today, as shown on the next slide, we will take you from the analytical complexity to the use of complex carbohydrates as an ingredient in a diet, to the point of looking at carbohydrate in foods, and finally, to the biological effects on the whole animal or human when complex carbohydrates are included as part of the diet. So to take us on to today's program, I'll turn this back to Marie, who will introduce our first speaker.
Thank you, Naomi, for setting the stage and telling us the exciting things we're going to hear about. And our first speaker, as noted, is Dr. Dave Luthria. So we will be turning the platform over to Dr. Luthria. He is a research chemist in the Methods and Application of Food Composition Laboratory, and today he will be speaking about dietary fiber analysis. So we turn it over to you, Dr. Luthria.
Thank you. Good afternoon, everyone. Thank you and for providing me an opportunity to present my research. The title of my presentation is Complexities Associated with Dietary Fiber Analysis. I can see that I'm a research chemist with MAFCL BHNRC group.
If you, Dr. Luthria, just click on the slide and then, yeah. Okay, there you
go. This slide shows the outline of my today's presentation. I will be discussing what are the complexities associated with fiber analysis. I will give you an overview of fiber analysis methods, and I will discuss about 17.16 and the two methods that we use in our lab for fiber content determination. I will briefly describe about our collaborative research results that we obtained with the UC Davis group, and I will end with the limitations of the current methodologies. The complexity stems from all the four aspects: fiber definition, structure, methods used for analysis, and impact of processing on fiber analysis. The dietary fiber exists in almost all natural plant-based products. The percentage of the dietary fiber varies in fruits, vegetables, and grains. The term dietary fiber was designed by Hipsley in 1953, saying a non-digestible constituents making up of plant cell walls. The definition has evolved as the time progressed. In 2001, American Association of Cereal Chemists defined fiber that are resistant to digestion and absorption in small intestine. This definition was further modified by Indian Institute of Medicine and National Academy of Sciences as a component of two things. Basically, it's a sum of dietary fiber and a functional fiber. The functional fiber can be actually modified fiber, it can be synthetic fiber, but it should have physiological health benefits. This definition was further modified by the Codex Committee of Nutrition and Food, and what they said is the fiber is a polymer of at least 10 monomeric units. And besides this, it should have other functions too. This definition was further modified by FDA, where they said it should have 3 monomeric units at least, and these should be not digested in small intestine, and it should have physiological health benefits. So basically, you can see that there is a constant change of definition. So if we have to quantify fiber, we have to understand what definitions do we follow, and this is one of the complexities associated with it. Now let me talk about chemical structure of the fiber. Fiber is a polysaccharide. It can contain anywhere from 3 to 15,000 units. And the monosaccharides can be varying from glucose, fructose, galactose, mannose. These are some of them. There can be multiple. Like if you look at a glucose structure, it has 6 carbon with 4 chiral centers. If we have 4 chiral centers, if I raise 2 raised to 4, that is 16 isomeric forms of just one simple glucose molecule. And the complexities with isomeric configurations with polymers will become enormous. The next thing is varying chain length. The chain length can be linear, branched, it can have different positions of linkage and chain composition. Therefore, fiber is the heterogeneous group of complex associated molecules. These are some of the terms that are used as fiber components: cellulose, hemicellulose, lignin, pectin. You can see that linkages vary in number. It varies in the branching. It varies in, besides this, they can have galacturonic acid, uronic acids. They can have phenolic compounds also, like ferulic acid attached to it. So fiber analysis is challenging because of that. This is a slide that I've just taken from one of the recent publications, which was published by Ian. You can see the complexities associated with the fiber. You can see how these are just some examples that they have picturized it in a very nice way. That fiber structure itself is complex, and when we do analysis, it becomes even more challenging. This slide shows the methodologies. These methodologies that we currently use it. You can see that different organizations have different methods, and these methods also have different variations based on this. And some of these methods still don't accumulate resistant starch, which is found in some of these foods. So you can see limitations associated with these methods. You can see number of methods that are being followed, and this gives these things. And these are just gravimetric methods. It doesn't give you any structural information about the fiber. What I will discuss in the next few slides is basically what we have been doing in our lab. We are doing 2 different methods. One of the method that is almost adopted universally, and it is labels that we generate of dietary fiber content, is using 991.43, which most of the databases are that have been used are there. And secondly, you can see that this is an automated method, but the only thing is we can do 6 parallel extractions and analysis at the same time. But there are a lot of manual steps that are involved, and there are challenges that we will see. Sometimes there are bubbles in the line which will have different amounts of solvent being buffed into it, and this will give variations in the results. There is insufficient validation data with this method, and there are time consuming because it requires a lot of evaporation, concentration, desalination, and a lot of these methods will require nitrogen and ash correction. We will be discussing more about it as we select some examples. This is a flow chart for the NCOM dietary fiber analysis that we used. What we do is basically analyze insoluble dietary fiber. We analyze soluble dietary fiber. We have methods which we can analyze total dietary fiber. And in most of these methods, when we get soluble dietary fibers, solubles, and waste. This also may contain fibers based on the definition that we use. This is a comparison of the 2 methods. As I told you before, AOAC 991.43 is the most widely used method, and this is used for documenting the fiber content of the food. But this method has limitations. As you can see that resistance starch is not calculated and low molecular dietary fibers are not calculated because they are considered as a waste in this. Whereas the method that we are comparing it out with 2017, which is a more modified method, the other important thing is the 991.43, the digestion occurs at 95 degrees centigrade, which is not physiological as compared to 37 degrees more physiological with the current method. These are some of the results. When we did one standard oat samples, we got results which were very similar to in both the cases, and these were also comparable with the literature information. Second thing, when we did it with corn, we saw the similar results. There were small differences, but the patterns were same. But the major thing that we saw the difference was in raw potato starch. This was a sample that Alan Smith is going to discuss more about it. You can see there was a huge difference between the two methods, and this was attributed to resistant starch, which is not estimated by 991.43 method. This slide shows you the collaborations that we have done with Dr. Carlito's group from UC Davis. And what we did is, this is a paper that has been just accepted for publication in Journal of Agriculture and Food Chemistry. Here, what we have done is we have moved from the gravimetric method, which is the weight differential method, to a method where we analyze monosaccharides. What are the monosaccharides present in these fibers? We have analyzed what are the different type of linkages that are present, and we have also analyzed what are the free saccharide contents in these food samples. This paper just has been accepted for publication in Journal of Agriculture and Food Chemistry. And this was done in collaboration with UC Davis Carlito's group, this entire research work. This is a fraction that is called soluble dietary fiber solubles. In this fraction, you can see that when you analyze it by MS, you can get triose, tetroses, stachyose, and other things. If we consider the FDA definition, then these all compounds has to be added to the fiber content which we get it through the gravimetric method, because these are all fiber molecules which may or may, we have to first find out whether they are digested in the small intestine, and secondly, we have to find out whether these have physiological functions to be considered into the definition. Now I come up to the next step is processing. If you take a fiber content of food, the raw food or the immature fruit or vegetable would have different fiber content than the matured food or the ripened fruit. So when we determine the numbers like a fiber and content of certain food, we have to say what was the maturity that it was taken from. And here I would show you that when we looked at mushroom, which can be consisted of a fruiting body and a stem, the fiber content in the stem was almost like 20 to 30% more than the fruiting body. Some people eat it with fruiting body, some with the stem, and some people take the stem out and just eat the fruiting body. So fiber content will vary based on what we consume. This is another example of the grain sample. This is a white wheat grain fractions. What we did is we separated the germ, the bran, and the refined flour. And we saw that, as we expected, the bran was the fraction that had the most fiber content. And when we say we did the fiber content of wheat, what type of wheat? Was it a refined wheat or whole wheat? We have to define those things in order to see what is the fiber content. It varies based on the part or how the grain has been fractionated. This slide actually shows, this is a work that I did in collaboration with Zenli as well as in Pamela Pearson's group. What they did is they collected the 3 corn samples and they processed it by steaming and boiling. From the crude fiber, the gravimetric method, we found that there were differences which were minor, but these samples have been submitted to UC Davis group to see if there are any compositional changes in this thing. This research is in progress, and we will soon get the results and get whether the processing impacts the monosaccharide level or the linkages level, or the free saccharides in these samples. So the challenge is right now, up till now, what we have been using or focusing is on gravimetric based methods. Our collaborations with UC Davis have gone one step forward where we do monosaccharide and linkages present and free saccharide analysis. Mike will be talking about gel permeation chromatography. So we want to take it to the next level where we can analyze what is the molecular weight of these different fibers, soluble, dietary soluble fibers as well as insoluble fibers, so that we can get more structural information about the fiber. And finally, we may have to use multiple techniques like high resolution nuclear magnetic electron microscopes and imaging to see how these polymer chains are packed and how do they impact the health functions. So in conclusion, I would like to say clarifying the fiber definition is important. Understanding what type of oligosaccharides are present, how they are digested and metabolized in intestine is present, because this is important if we follow the fiber definitions. And besides this, I have not gone into the details. Some of the fibers like polysaccharides are N-acetylglucosamine units, especially chitin-rich foods, which are found in mushrooms. They may contain nitrogen interferences, and we have to find out the way. Basically, we may have to do a couple of combination of multiple techniques to identify what is fiber and what is the nitrogen from the protein. And finally, as I showed you, the sample preparation, sampling stage, sample processing, particle size, these all impact the particle fiber content determination. And we have to use appropriate methods for fiber content. And lastly, I would like to thank my group colleagues, Dr. Fakir, Tariq, Raghu Kota, and Shami Samia for their help in carrying out these analysis. I am thankful to the USDA team members for their help in actually formulating this process to find out more detailed analysis of fiber. The UC Davis group for giving us structural information and other things, and members from NCOM, because we did, when we got this new technology, we did comparative studies with NCOM as well as we did some nitrogen content analysis from with other groups. So I would like to thank all of these people for the help in carrying out analysis. Thank you.
Wonderful. Thank you, Dr. Lothria. Seems the more we learn, the less we know, especially about fiber in this case. But I'd like to introduce our second speaker, Dr. Mike Bukowski. And Dr. Bukowski, if you'd like to join us on webcam, that'd be great. He is a research chemist at Beltsville, and today he'll be talking about the future plans to advance the analysis of carbohydrates following on Dr. Lothria. So, Dr. Bukowski.
Yeah. So, presumably everyone can see my slides right now. And I just want to give a brief bit of background on myself. I've been here about a year in Beltsville, but before that, I was at the Grand Forks Human Nutrition Research Center running the Nutritional Analytic Core. And where I spent a lot of my time was in developing analyses for lipids, lipidomics, and biomarkers. But prior to that, I also did work in polymers and gels. And so in coming to this problem as for carbohydrate analysis, I would like to move kind of, as the title says, beyond functional definitions of dietary fiber. One of the highlights of working in Beltsville is that we've got a wonderful public-facing product a lot of you may be familiar with. This is Food Data Central, which combines the standard reference legacy foods you probably may be familiar with survey foods at FNDDS, branded foods, and experimental foods is off the side of the screen, and a new addition, the foundation foods, which are things that are have limited processing. They're things that contain basically one major ingredient. But I'm just going to go and put beans in here because beans are a wonderful source of dietary fiber. And if you pull this up, the SR, this is a nice SR entry for beans. And we can see we have our components here, our approximates, right? Protein, lipid, ash, and carbohydrate. You see carbohydrate by difference, right? Carbohydrate is measured as that which is not protein, lipid, or ash, or water. And if we're really lucky, we get dietary fiber, total dietary fiber present, and that's by the enzymatic method that Dr. Lothria presented previously. But if we dig a little bit further, let's grab approximate, let's take a look at just total fat. You can dig into this and you get all the fatty acids coming out of it, right? And the thing is, each component in here is got some nutritional salience. You have chain length, you have desaturation, you have double bond position, you have double bond isomerization. So you can tell if something's a polyunsaturated, a long chain polyunsaturated, omega-3, a saturated fatty acid, or even a E or Z, right? You can tell if something's a trans fatty acid, all by language you're very familiar with by going to Food Data Central. And what this is it's a special vocabulary for discussing lipids in this nutritional sense. And the question is there something we could do comparable here for carbohydrates and total dietary fiber? Well, other than the definition that we have to this point, which is dietary fiber does not appear to react with alpha amylase or alpha glucosidase to generate glucose. And so from the previous 20 minutes or so, you've seen this method, which is basically the 2017.16, and this is the total integrated dietary fiber method. And these are the all enzymatic conditions. If you take nothing else home from this particular slide, it's that this entire definition of dietary fiber is based essentially on enzyme kinetics. So it's going to be variable by activity of the enzymes. It's going to be variable by temperature. It's going to be variable by how long you keep it in and keep incubate your reaction. Additionally, these definitions are based on the ability to precipitate an ethanol. There are lots of things that can interfere with precipitation in ethanol or promote precipitation in ethanol that would create variability in this method, right? The one thing this definitely has as a strength is that these compounds, this can be done on benchtop. Anybody can go buy these things and perform this experiment. But if you take a look at dietary fiber, right, the dietary guidelines since 2005 have said that dietary fiber is an underconsumed nutrient of public health, and a lot of people in the industry have tried to meet that need for dietary fiber, about 12.5 grams per 1,000 kilocalories or so. And you can meet that in a variety of ways. You can do this with supplements, you can do this with functional foods. And there are a couple ways of approaching this problem. So for supplements, you have fructooligosaccharides. Basically, everything I have underlined here is something that would pass for soluble dietary fiber following that AOAC method on the previous page. But they do it through different mechanisms, right? Fructooligosaccharides, pectin, agar, tapioca starch, or even modified wheat starch, where something has been taken from a natural product, been modified chemically to make it resistant to digestion. Right? And so if we just blow up a few of these, the fructooligosaccharides, basically you have the wrong kind of monomer. You have a fructose instead of a glucose. So alpha amylase is not going to interact with this. It's not going to bind it and cleave it, right? Similarly, pectin, you've got the galactonic acid, which also has the ability to have modifications here. Other oligosaccharides can be hanging off the backbone, or you can have acylation or methylation. All of these variations are going to impact how this interacts, but additionally, it's not going to react with our digestive enzymes because it's not an alpha glucose based chain. Similarly, agar. We have a repeat unit in agar. This is an extract from seaweed that's used in a lot of things in our diet, and it just has a different and weird repeat unit, right? With this dehydrated bridgehead in one of the monomers. Then you can take a look at, this would be tapioca starch. Looks like it should work just fine. It's a glucose, it's alpha bond here at the anomeric bridgehead here, anomeric head here. But because it's type 3 starch, it actually forms a three-dimensional structure that makes it resistant to digestion. And finally, we have the modified version where we have, in this case, I'm showing a phosphate modified one where they add, phosphorus trichloride, and it just inserts phosphate groups, bridging and making this a viscous solution. So as previously described, the equivalent of the FAMES data that we have in Food Data Central would be this monomeric composition. And we have the good fortune to be working with Dr. Carlito Labrea in his lab at UC Davis, and they do a series of steps. They do a derivatization, which is often methylation. They do a chain cleavage, and that allows them to basically take everything in this mixture here, and they can sort it out and say, okay, well, those are terminal, those are bridging, and those are glucoses that are in the chain. And then they can go through and do the rest for the rest of these components in the mixture. But you see what we lost here. We went from having all this individual material, all this individual structural information, to basically having a bunch of monomers, right? So this is informative, and this is definitely at the level of what we did for fatty acids for a very long time. But with the advent of lipidomics, things got more sophisticated. With the advent of some of the glycomics in nutrition, things are going to get more sophisticated. So we have to have a slightly different language with which we discuss this, right? So is just monomer composition appropriate? Well, not really, because we have things like viscosity and fermentability that impact in this case, this is dietary fiber and its cholesterol lowering abilities. It's been known for a long time that viscosity has an impact on the ability of dietary fiber to have healthful benefits. But this is, think of it this way from a material science perspective, this is chain link, this is cross-linking, this is branching, right? Those are the parameters that are going to determine viscosity. You know, this is a recent study where inulin with different chain links was used to have differential impact on endotoxemia. We have another paper here where you have, this is pectin from 2 different varietals. Presumably there's modifications on that backbone, on the acid backbone, that result in different functionality. And even this recent paper in Cell Host, that they basically took 2 of the most common fibers people use, fructooligosaccharides and arabinoxylate, psyllium husk fiber, right? And they compare the 2 in terms of the distinctive personal changes people have in their microbiomes as a result. So, you know, it turns out the structural stuff is very important. So we're going to have some other nutritional characteristics we consider, right? Because this is a polymer, not a discrete compound. So we'll have primary, secondary, tertiary, quaternary characteristics. And I'm going to pick up the pace here because I only have 10 minutes. But by bringing this all out here, we have primary characteristics like monomers, secondary characteristics like polydispersity, that's chain length and degrees of branching. Then we have tertiary things like structure, 3-dimensional structure, which is something like type 3 dietary fiber or type 3 resistant starch or type 4 resistant starch, where you have interactions within the chain or between the chain and other things within the matrix. Our goal is to build a way of taking these mixtures of dietary fiber and separating them out by other material qualities. So for example, chain length impacts hydrodynamic volume. Each one of these things has the same number of dots in it. Each one of these has the same number of monomers, but because of branching, or in this case, 3-dimensional structure, they're all going to elute at different times, come out at different times in gel permeation chromatography. We can take this then and bring this over to HPLC, high resolution HPLC mass spec, and we'll be able to, again, go through and look at our monomers for which we have, or monomers or dimers for which we have standards, but then we'll be able to, since it's high resolution mass spectrometry, pull out actual chain lengths that are otherwise lost in size exclusion chromatography, because mass spec has superior mass resolution. Similarly, where things overlap, these are 2 different kinds of dietary fiber, they overlap, we'll be able to tease them apart because they will have different mass regimes. And then when we have cases where we have bridging and functionality, we'll be able to see different mass patterns. Right, so the goal here is to get more sophisticated in our ability to express what the dietary fiber is. We still won't be away from enzymes, but in this case, we'll have moved beyond enzymes as the quantitative metric to more of a qualitative metric. So we can't do, for example, you know, amylase and you know, alpha and beta bonds at the anomeric carbon. We can't really do that right now by mass spectrometry, but enzymes can. And if we don't care about the kinetics, we could take as long as we need. So anyway, what does this mean for nutrition as I hand it off to Alan here? We're going to have a more sophisticated vocabulary. I can't tell you at this point which of these vocabulary words on the screen here is going to be critical and which combination of them will be critical. But we need to have methods that address the sophisticated and you know, difficult problem of treating something we all need as a material, right? And not just as a food, but as a material science component, right? So the goal out of this is to be able to analyze whole foods or meals so we can get fiber and put it in the right bins, right? Why is this a resistant starch? Because then, since we're eating for basically 1 billion microbes, we have a structural characterization that can feed into the metagenomic analysis to create structure function relationships. So with that, I'm going to hand this off to Alan. I've had my 12 minutes, so thank you.
Great, thank you, Dr. Bukowski. So as mentioned, our next speaker is Dr. Alan Smith, and Dr. Smith is a research virologist in the Genomics and Immunology Laboratory, and he will be talking today about his work on resistant starch and links there to animal gut health. So Dr. Smith.
Thank you very much for the invitation today to be able to share some of our work. And we're going to shift a little bit from a more analytical stuff to more of a what's going on with resistant starches in a biological setting. So I have nothing to disclose. As most of you are aware, there are three broad classes of starches. There's rapidly digested starch, which is digested in the mouth and small intestine and rapidly releases glucose. There's slowly digested starches, which are digested in the small intestine and slowly release glucose. And then there is resistant starch, which is basically not digested in the small intestine, makes it to the large intestine unchanged, and is fermented to short-chain fatty acids and other metabolites by commensal bacteria. As Dr. Bukowski talked about earlier, there are different types of resistant starches. There's resistant starch 1, which is physically inaccessible starch granules. There's resistant starch 2, which is ungelatinized resistant granules that is crystalline in nature. There are RS3s, which are retrograded starches. This is a result of cooking and cooling, such as with potatoes. You have what would be considered unnatural resistant starches because they're chemically modified starches that are cross-linked. And then you have type 5 resistant starches, which are amylose lipid complexes. So starch granules are composed of a combination of amylose and amylopectin chains that can vary in their ratio depending on the starch. They fold up into increasingly more complex structures, such that you eventually end up with a starch grain. And this starch grain, for example, with RS2 starch, can have alternating layers of crystalline structure and amorphous structure that can affect the ability of the commensal bacteria to act on it. So commensal bacteria break down the starches in the large intestine and in the cecum, for example, in mice. And various bacterial strains possess different sets of genes encoding carbohydrate active enzymes that degrade a wide variety of complex carbohydrates. And if you look at the commensal population in your gut as a whole, there are literally hundreds of these genes encoding different enzymes for breaking down starch. So commensal bacteria can be divided into three basic categories: primary degraders, which can act on an intact starch granule and can start to break down the outer surface of the starch granule, that then allows secondary degraders to get access to the starch granule to further break it down. And then you have cross feeders, which can take the metabolites resulting from primary and secondary degraders and use those to create additional secondary metabolites. So ideally, if you're going to do starch studies, you would do them in humans because it produces the most relevant data. However, there are limitations to human studies. It's hard to rigorously control a diet for human subjects for extended periods of time, and you can't do infectious disease challenges. So animal studies are the next best alternative. And in our lab, we have conducted studies in mice looking at the effects of consumption of resistant potato starch on the microbiome, host gene expression, and resistance to a gastrointestinal bacterial challenge. So then this raises the question of what is the best basal diet for animal studies? Again, ideally, you would be able to mimic a typical American diet. And so what's happened in the past is with traditional nutrition-based studies have used diets prepared from purified ingredients formulated in collaboration with the American Institute of Nutrition. So the original formulation came out in 1976 and was called AIN 76A. That was subsequently modified in 1993 to produce AIN 93G and M. And these diets were subsequently modified by many different labs, especially those interested in doing high fat studies in obesity and diabetes, where they increase the amount of fat to very high levels using either lard or anhydrous milk fat. But again, these diets do not represent what the average American consumes. So we instead took a different approach and used a diet developed by Corey Heinz and his colleagues at Utah State University that's called the Total Western Diet. And this diet has the advantage of that it is based on NHANES data. And so the 50th percentile daily intake levels for macro and micronutrients from NHANES data was used to formulate this. Fewer calories are from protein and carbohydrates. It has double the fat, but importantly, the fat is from a variety of sources. It has fewer complex carbohydrates and twice the level of simple sugars. And also importantly, it has less micronutrients and vitamins and more sodium compared to the original AIN 93 diet. So this is our basic experimental design. We placed mice on a total Western diet for 6 weeks. We then switched them to the total Western diet containing 0, 2, 5, or 10% RPS for 3 weeks, and then we collected samples for analysis. So we collected cecal contents, and again, in mice, the cecum is the primary site of fermentation, and so we looked at short-chain fatty acid productions in cecal contents, and we didn't see any changes in total acetic or propionic acids, but we did see an increase in butyric acid, which has been seen by others as well. And interestingly, we saw a decrease in isobutyric, isovaleric, and valeric acids, and this suggested that there was a shift from fermentation of proteins to fermentation of carbohydrates. We then looked at the microbiota present in the cecal contents, and the first question we asked was what's going on with the alpha diversity. And alpha diversity looks at the amount of diversity of various bacteria within a given group. So we saw that as you increase the amount of RPS in the diet, the alpha diversity decreased, and this was pretty much uniform in 4 different slightly measures of alpha diversity. And again, there have been multiple studies in mice, swine, and humans that have observed similar decreases in alpha diversity in response to various resistant starch and fibers, suggesting that this may be a rather universal phenomenon. We then looked at the beta diversity of the microbiota in the cecal contents, and this looks at how much difference is there between groups. So as you can see, the 3 groups that were fed the different levels of the raw potato starch were significantly separated from the animals on the basal diet. But even within the group that received RPS in the diet, you could see distinct separation based on the amount of RPS in the diet. So then we drilled down to look at what was happening with individual genera in the cecal contents, and we came across a rather surprising result, and that was shown here in light purple is Lactobacillus NKA136 group, which went from basically 10% of the total microbiota present to 50% as you went from 0 to 10% RPS in the diet. So this became the dominant species in the microbiota of the RPS 10% fed mice. And so this more than likely helped contribute to the decrease in diversity that was seen, since you have one species that is really taking over. So then we looked at the effect of feeding RPS on gene expression in 3 different tissues by next generation sequencing. And these are PCA plots, which again show the degree of difference between groups. And as you can see, as we saw with the microbiota, gene expression was changed in a dose-dependent manner based on the amount of RPS in the diet. This was less prevalent in the proximal colon and even less so in the distal colon. In addition, we saw that the greatest number of changes occurred in mice receiving 10% RPS, and there was not nearly as many in the 2% RPS, and there was significantly more genes upregulated than downregulated in all 3 tissues. Now, I don't really have time to go into all the different changes that were occurring, but one thing that was universal between the cecum, proximal, and distal colon was that they had genes associated with antibacterial, antiviral, antiparasitic immune responses that were upregulated, indicating that RPS may be priming the immune system for resistance to infection. So we wanted to actually test that out. So there's a very convenient model using Citrobacter rodentium, which is a gram-negative bacteria that causes infectious colitis in mice. And essentially, it's E. coli that infects mice. It shares many characteristics with human enteropathogenic E. coli. And it, after oral inoculation, it initially colonizes the cecum, and then it starts to colonize the distal colon at 3 to 4 days later, with peak infection occurring between days 6 and 10, and it's usually cleared by day 21. It induces a potent Th1, Th17 immune response, and it has been a very useful model for studying infectious colitis caused by foodborne pathogens. So we just modified our basic experimental design a little bit. So again, mice are placed on the total Western diet for 6 weeks, followed by diets containing RPS, and then 3 weeks later, they were infected with Citrobacter rodentium, and we collected samples, fecal samples at days 4 and 6 after infection, and cecal contents and tissues at day 12 post-infection. So again, we looked at alpha diversity, and as we saw before, in uninfected mice, the alpha diversity decreased with increasing amounts of RPS, but interestingly, in cecal contents, this effect was blunted by infection. When we looked at feces obtained at day 6 post-infection, we did not see this effect wasn't nearly as pronounced as it was in the cecal contents, in that some measures of alpha diversity did show some effect, while others did not. So then we looked at beta diversity in the cecal contents, and we also looked in the fecal contents too, but I'm only going to show the results from the cecal contents because they were very similar between the two. So as you can see here, all the animals that were infected grouped together and away from all the animals that were uninfected. And even within the groups that were either infected or uninfected, there was a dose-dependent, as RPS increased in the diet, there was a separation of the various groups. So again, we drilled down to look at what was going on with individual genera. And again, just focusing on what was happening with the Lactose Biraceae NKA 136 group. Again, in uninfected mice, we saw this stair-step increase to where about 50% of the genome was occupied by this genera by the time you get up to 10% raw potato starch in the diet. Interestingly, if you look at what happened in the infected mice, there was this pretty much the same jump with 2% raw potato starch in the diet, but after that, it really didn't increase all that much more. And again, this is probably related back to the blunting effect of the infection on alpha diversity. So then we wanted to take a look at what was going on as far as CR infection directly in the host. So as I stated before, the cecum is initially colonized, and then starting about day 3, 4, you start to see colonization of the distal colon, and that is the primary site of infection from there on out. So we looked to see what, if any, effect there was on RPS on the efficiency of infection of these animals, expecting that RPS would improve things based on our previous gene expression studies. And in fact, we saw the exact opposite. So if you look at the animals on the basal diet, 6 of 16 animals were not productively infected, while all the animals receiving any level of resistant potato starch saw 100% take of the infection. So it would appear that the efficiency of infection was improved by having RPS in the diet. In addition, we saw that there was an increased fecal secretion of Citrobacter rodentium, and this again was a dose-dependent increase with the greatest effect seen at 10% RPS. Again, suggesting that the raw potatoes. So then we looked out at day 12 post-infection at what was going on in the colon tissue. At this point, you have C. rodentium attached to the mucosa surface, and it is about this time that the host is pushing the bacteria off the mucosa surface so that it can be cleared from the host. And again, to our surprise, we saw that there was an increased amount of Citrobacter rodentium attached to colon tissue at day 12 post-infection compared to the other groups. So again, it appeared that RPS was increasing its ability to attach to the mucosa surface. Since Citrobacter rodentium is known to cause damage to the mucosa tissue, and it's related to the amount of bacteria adhered to the mucosa surface, we wanted to see if there was any difference in the amount of pathology in the 10% RPS group compared to the animals on the basal diet. And that is exactly what we saw. So you had increased colonization of the tissue, you had increased histopathology in the 10% RPS group compared to the animals on the basal diet. We then looked at, again, gene expression again by next generation sequencing. And as we saw with the microbiome, you saw a distinct separation of animals that were infected from those that were uninfected. But unlike what we saw in the microbiota, there wasn't that much separation between the various doses of RPS in the diet, so that the main effect was due to infection. So again, I don't have time to go into a ton of different changes because we saw a huge number of changes induced by, especially by infection. But shown here is a heat map where green indicates upregulation, red indicates downregulation, and if it has a letter, indicates that it was significantly different. So in this section is results from the cecum and there's this section from the distal colon, 12 days post-infection, and each column is comparing infected versus uninfected on a basal diet. So here you have 0, 2, 5, and 10, 0, 2, 5, and 10. And what you can see is that if you look at a gene like gamma interferon, which is important for clearance of Citrobacter rodentium, it was increased both in cecum and distal colon. Interestingly, though, another gene important for clearance, IL-12A, was basically unchanged in the cecum, but was significantly downregulated in the distal colon, and that was highest at the 10% RPS dose. Other genes important for clearance, such as IDO1 and INOS2, were upregulated in both the cecum and in the distal colon. And we saw IL-22, which is important for barrier maintenance, was increased, while the antagonist IL-22RA2 was decreased in both the cecum and in the colon in a dose-dependent manner. And again, it was highest in 10% RPS group. And what this suggests is that it wanted to downregulate the antagonist of IL-22, so there would be more IL-22 to be active. And the other thing that we observed was that there was a decrease in inflammatory markers in the cecum versus the distal colon. And this is not surprised, since the cecum is already clearing the infection at this point, and it's still active in the distal colon. And lastly, we did see some downregulation of genes associated with epithelium maintenance and tissue repair. And again, it was most prevalent in the 10% RPS group. And lastly, we saw something that we found very intriguing and interesting, in that mice fed the 10% RPS diet and infected with Citrobacter rodentium had significant downregulation of retinol metabolism in the distal colon. So shown here is a KEGG diagram, and wherever you see a red star, this indicates that that gene was downregulated in the 10% RPS infected group. Now, VA is essentially for differentiation of gut epithelial cells, T cells, B cells, and macrophages, and in the form of retinoic acid is required for the IL-17 dependent immune response to Citrobacter rodentium. And we and others have shown that vitamin A deficient mice infected with Citrobacter rodentium have enhanced colonization and increased pathology. And so this offers a possible mechanism by where the 10% RPS group might be having these increased colonization and pathology that we observed. And lastly, I'd just like to point out one other possible mechanism for what might be going on early in infection when we saw increased efficiency, and that is as the commensal bacteria ferment RPS or other fibers, it produces sugars. And now monosaccharides are actually preferred by C. rodentium and E. coli-like organisms, and actually can promote their growth. In addition to which, butyrate, 1,2-propane-diol, and succinate are all compounds that are produced from fermentation of resistant starches and fibers, and all three of these compounds are known to bind to Citrobacter genome and enhance expression of virulence genes associated with colonization and pathology. And lastly, I'd just like to point out that there has been a study done where animals were fed a diet essentially devoid in fiber, and they also saw that you had increased susceptibility to C. rodentium and increased pathology and colonization. And in this case, what was going on was that since there was no fiber or resistant starch in the diet, these animals, these bacteria, had nothing to feed on. So what they did was they attacked the mucus layer, using that as food, thus degrading the mucus layer and making the host more susceptible to binding of Citrobacter rodentium, induction of pathology. And lastly, I'd just like to say that other groups have shown that pectin was actually protective against Citrobacter infection, while another group, using a high-fat diet with cellulose as the fiber content, saw a decreased colonization, just as we did, and they found that inulin reversed this, as did a chow diet. So again, there's an example where an increase of resistance of a type of fiber or a complex mixture of fibers led to an increase in the ability of Citrobacter rodentium to colonize. And then lastly, I just want to conclude with consumption of raw potato starch can significantly modify the microbiome of mice, even at low concentrations. The combination of dietary RPS and C. rodentium infection produced unique changes to the cecal and fecal microbiome. It increased efficiency of colonization and also increased the amount of Citrobacter rodentium associated with the tissue, and also increased the amount of pathology that we observed. We saw large numbers of gene changes associated with both diet and infection in the cecum and colon. And we believe that this raises an important question of how consumption of different types of fiber or resistant starches can impact susceptibility to a common class of human pathogens. And last but not least, I'd like to thank members of my lab, Harry Dawson, Celine Chen, and Lumei Chung, whose efforts were instrumental in coming up with this data, and also collaborators at Utah State University, Robert Ward and Corey Hines. Thank you very much.
Wonderful. Thank you, Dr. Smith. And next we have joining us is Dr. Pamela Person. And Pamela, if you'd like to turn on your webcam. So Dr. Person is a nutritionist in the Methods and Applications of Food Composition Laboratory. And today she'll be discussing carbohydrates in both raw and processed foods. So I will share my screen for you, Dr. Person.
Can you hear me?
Yep, we can hear you loud and clear.
Okay.
All right.
Thank you very much. And thank you for on 2 studies at BHNRC that focus knowledge that corn is the 3rd most highly consumed vegetable behind potatoes and tomatoes. And it's also a significant source of carbohydrate. On order per 100 grams, about 20 grams of carbohydrate and 70 to 75 grams of water. Hence these targeted studies. Next slide, please. Thank you, and you can go through all the bullets. Thanks. So we've been generating carbohydrate data for many decades, including sugars and starch and dietary fiber. However, more recently, and building on the presentations, the preceding presentations, we've been evaluating and optimizing the methods that quickly and accurately measure total dietary fiber and its major fractions, because as we know, the definition of fiber is always changing. We're looking at characterization of structural information, such as the alpha and beta glycosidic linkages that Dave Luthria mentioned, of complex carbohydrates in collaboration with UC Davis, as well with UC Davis, oligosaccharide measurements, and in particular, and presented or will be presented in Food Data Central, those for wheat, rye, barley, beans, nuts, many vegetables, and even the plant-based milks, in quotes. And all with the express purpose of supporting research, but also generating more detailed carbohydrate data for Food Data Central. We're also looking at how the content and structural alterations of these complex carbohydrates after processing and cooking can affect bioaccessibility, bioavailability, and utilization in vitro or in animal models of these carbohydrates. So in particular, we're going to focus on 2 studies, both managed by Dr. Shenli Wu of our Food and Nutrition Research team here in Beltsville, and also with Dr. Monica Wendt, who's been involved in this. And finally, in a separate study with colleagues in China focused on the same research. Next slide, please. So we know that the background of carbohydrates should include the effects of food processing and cooking on their carbohydrate profiles. We know that carbohydrates are major macronutrients in plants, and that they're consumed after industrial processing and home cooking. We also know that carbohydrates can be altered quantitatively and qualitatively during food processing and cooking in a number of ways. These include loss during processing, so for example, during washing and cutting or removal of certain parts, during gelatinization, which refers to the disruption of crystalline structure within the starch granules, and this occurs upon heating in the presence of water. That facilitates starch degradation as recrystallization of the gelatinized starch as a result of cooking and then cooling. and that may reduce the digestibility. It's also impacted by food processing. It can also break down chemical bonds, alter the fiber matrix, change the hydration properties of carbohydrates. All of these can be impacted by processing and cooking. And this ultimately impacts the digestibility and absorption of the carbohydrates and their utilization by the human body and by the gut microbiota, which ultimately determines their physiological functions and health benefits. Next slide, please. Next slide, please. Thank you. This is a recently published study that looks at the effects of two different cooking methods on the carbohydrate profile in sweet corn. And in this study design, we have two varieties of sweet corn, yellow and bicolor, and they were purchased in bulk overseas at a grocery store. Each variety was divided into three groups. One is raw, the other two boiled, and the third steamed. The raw in liquid nitrogen and freeze-dried immediately so that we preserve the samples. Different sugars and sugar alcohols, oligosaccharides, starch, resistant starch, total water-soluble carbohydrates, and dietary fiber were analyzed in these corn samples, both raw and in the two cooked samples. Details on these methods are in the paper at the bottom right of your screen, recently published. Next slide, please. So, next slide. Slide shows that the effects of cooking on the carbohydrates of sweet corn, and these are data on individual sugars and sugar alcohols. There were 4 simple sugars that were identified in both varieties: maltose, sucrose, glucose, and fructose, with sucrose being the predominant sugar in both. The other 3 individual sugars existed at extremely low concentrations, and maltose wasn't even detected in the yellow sweet corn. 2 sugar alcohols, sorbitol and mannitol, were also identified and measured. The 2 cooking methods did not alter sucrose and mannitol significantly, but it did cause reduction of other sugars or sugar alcohols. Nevertheless, if you look at all of the concentrations of the sugars and sugar alcohols, cooking, at least in this study, did not change the overall sweetness of the corn or those individual sugars that would contribute to sweetness. The difference between the 2 studies that I'm presenting to you is one was in collaboration with Chinese scientists, Chinese colleagues, and those studies were retail samples of corn. In our study, and a lot of the data aligns with the study in the international study, was actually one of the first of its kind where our scientists went to the field, particularly Dr. Wu, and actually handpicked the corn and enough to complete the entire study raw and cooked. So it's following the changes in carbohydrates from the actual original sample all the way through commercial processing. And we do have an agreement and are working with the University of Maryland that has the facilities to simulate commercial freezing and canning, etc. So we feel like we're adequately representing the entire process from field to finished product. Next slide, please. I'll just start as you're moving to the next slide. The 2 cooking methods, boiling and steaming, really, as I said, did not change the starch and total water-soluble carbohydrates. And I know that Dr. Luthria talked about this in his presentation. Total water-soluble carbohydrates were measured because That's what gives sweet corn a good mouthfeel, that and sugars. The combination of endosperm water-soluble carbohydrates and sugar content is a very important determining factor of the sweet corn quality. And I know from a nutritional standpoint, we don't look at that, but it is something to be considered. The starch that's measured is total starch. That is digestible starch and resistant starch. So during the processing of the original samples, we tried to achieve total gelatinization. And again, in the paper referenced at the bottom, it'll give details. And then in the cooked corn, we therefore saw no change in starch, but the resistant starch appeared to have converted to digestible starch. The water-soluble carbohydrates in this study included simple sugars, oligosaccharides, in some cases, but not all, fructans and starch. Next slide, please. So in most plant foods, total dietary fiber largely consists of high molecular weight dietary fiber, resistant starch, and non-digestible oligosaccharides. In our study, these fractions were measured individually. So no oligosaccharides were detected in raw or cooked samples. Therefore, the TDF or fiber was calculated as and the resistant starch that were measured. And you can see that in panel B on the right. And as you can see, the cooking did not change the high molecular weight dietary fiber, but significantly reduced resistant starch. Food processing or cooking techniques may affect both the gelatinization and the retrogradation processes of starch, which can result in the increase or decrease in the resistant starch from those found in the original foods. So these data suggest that the increase in digestion through gelatinization may be the primary reason for the loss of resistant starch after cooking. Next slide, please. I will conclude the information on our corn studies, which are ongoing, and just mention a few other activities within this team. We are looking at dietary fibers and other foods commonly found in the U.S. food supply for analysis, and most recently a focus on foods that may be included in the Mediterranean diet. So I've listed here some of those fractions, and I'd mentioned that in the beginning. We're interested in these fractions. Insoluble dietary fibers, cellulose, lignin, hemicellulose, pectin, and soluble fibers listed below, including inulin and the oligosaccharides. And then the non-digestible oligosaccharides, the trioses and tetroses. So we'll continue expanding that research, and we'll go to the last slide, please. I just wanted to show you an example of some of the oligosaccharide data that we've generated, and I believe this is now included for these two foods in Foundation Foods and FDC. And these are the oligosaccharides, Stachyos, verbascose in the plant-based milk alternatives, which are really trending now, as you might imagine. So we're really exploring these foods. The next slide, please. I'd like to acknowledge Dr. Shenli Wu and Dr. Monica Wendt, and also not included here, Dr. Dave Luthria, who really contributed a lot to this research. At University of Maryland, Dr. Lucy Yu and Holly Childs, and then our colleagues at the University of Macau for the international study that I discussed. Thank you very much.
Great, thank you, Dr. Person. And we have one more speaker. Our final speaker is Dr. Tom Wang. And Dr. Wang, if you'd like to join us. So Dr. Wang is a research leader in the Diet Genomics and Immunology Laboratory at Beltsville. And today he'll speak to us about resistant starch from rice and effects on obesity and the gut microbiome. So welcome, Dr. Wang.
Thank you. The advantages of being the last is prolonged agony. So I'm gonna talk about the effect of cooked rice. So I kind of round everything up and then try to talk about a food. These are excerpts from two of our previous publications shown over here. And hopefully, if you have a chance, you can go into those publications and look at it in more details. So my disclaimer, disclosures, I work for USDA, but I'm also on the live micro subcommittee for IAFNS. This is kind of the kind of question that I ask to frame my research. So, I mean, from a nutrition point of view, we want to know what's good for you, when it's good for you, where it's good for you, why it's or how it's good for you, and how much it's good for you, as well as who it's good for. With the advances in microbiome, I kind of affectionately call our research is from farm to poop. Rice is an interesting grain because, well, one of the things is we don't eat that much. US don't consume that much rice, but if you look at worldwide, it's ranked number 3, right next to wheat and corn. However, economically, US is doing pretty well. We actually is the number 5 producer or exporter for rice. And you can see over here that there's a lot of room to grow if we can help the American farmer to sell more rice. But the other thing is scientifically is rice have also have some interesting property. One of them is if you cook rice, it will generate structural changes in the starch, and then you generate resistant starch, and in this case, it's called RS3. So there's a lot of work that is looking at it, try to improve processing or breeding and things like that to up the resistant starch. But why is that? Sadly. Because in the literature, there's several beneficial effects of resistant starch has been reported. Basically everything under the sun, including prevention of diabetes, regulation of lipid metabolism, improvement in insulin resistance, decrease in intestinal inflammation, as well as reduction of colorectal cancer risk. So almost everything under the sun. And it is thought that maybe generation of short-chain fatty acids as well changes in the microbiome may be the one that changes things. However, there's still a lot of questions regarding resistant starch research. For example, if resistant starch introduced as a whole food, such as cooked rice, for example, does it have a very similar effect as compared to the isolated resistant starch? Even though there's a lot of study out there looking at mechanism, but the precise mechanism it is not known, as well as if it's in a whole food, what would happen? Another thing is, what are the concentration of resistant starch in whole food required to elicit some kind of biological effect? For example, like changes in body weight, lipid metabolism, short-chain fatty acid, and so on and so forth. Now, when you develop a food, a new varietal, I think it's important to at least validate the biological effect, because if you can say I'm going to increase something for 1,000-fold, but it didn't do anything, you may not mean anything. And so with that kind of a list of questions, we thought rice is a very simplest system to address these questions because it's mostly starch. So this is working with some of the folks in ARS in Del Bumper International Rice Center, and we decided to look at 3 varietals. There's a low resistant starch one, there's the one we call the medium resistant starch and high resistant starch, and it's go from 0.1 to all the way up to 8.6. We try to use this low resistant starch one as kind of more like a matrix control. So the design is a 2x3 design. We have 10% or 39% fat matrix, and the starch is come in the form of cooked rice. This is using a C57 black mice and with 410 gram per kilogram of starch in the diet. So this is an 8-week feeding study. The thing that we found is that it didn't do much on the body weight. So, we fed a high-fat diet. We thought, well, great, let's see if it lowered the body weight, but it didn't do much to the body weight. But what we found is that the adipose weight as well as the adipocyte sizes seem to be attenuated by increasing amount of resistant starch. Shown over here is staining. It's called it's oil red staining of the adipose tissue. The other thing we look at is try to look at molecular markers in the adipose tissue. And what we found is that lipid metabolism, as well as inflammation-related markers seem to be affected by resistant starch. For example, you have the leptin get attenuated, LPL, as well as ATGL, which is adipose triglyceride lipase, hormone-sensitive lipase, and two of the inflammatory marker, the MCP1, also called CCL2, and IL-6. So somehow the high resistance starch is very effective in blocking the adipose tissue related event. To a certain extent, a medium level medium resistance starch rice also did something. We also look at the effect on plasma and as well as liver, and it turned out that in the plasma it seems to mimic what's in the adipose tissue, that you have the resistant starch attenuating the increase of leptin in the plasma. What's interesting is that only the high resistant starch seem to affect the liver triglyceride level, as well as increasing the triglyceride content in the feces. We look at quite a few markers in the colonic tissue, but end up kind of depressingly not finding any changes. It's kind of surprising that we thought maybe there was something changes. But we did find that the short chain fatty acid seemed to change significantly in a high resistance starch diet. You can see over here acetate, propionate, butyrate, as well as changes in the gut pH level. And of course, the obligatory gut microbiome research, as you can see over here, you can see these changes in progressively from low to high concentration of resistant starch. One of the things I need to point out is we did not see any big changes between different fat level. This over here is very similar to what Dr. Smith have shown, that there seem to be changes, actually lower diversity levels. So somehow by eating these resistant starch, the gut microbiome seems to be less diverse, or the bacteria tend to be less diverse. We did a regression analysis on the between the bacteria and the production of short chain fatty acid, and we found that the two family S24-7 and this is called bacterial daisies. They seem to correlate with the concentration of generation of all these short chain fatty acids. So, to summarize the working model for for us, and as far as the cooked rice, is that we did see a concentration dependent effect at the 1% level. You see attenuation of adipose weight changes, as well as changes in gut microbiome, and lipid may be playing a role in here somewhere. As you go up to a higher level, you see changes in short-chain fatty acid. You see effect on the liver triglyceride, as well as increase in triglyceride excretion. So somehow, if you consume the rice, consume the resistant starch, in this case RS3, as quick rice, all these changes can occur. So how about some idea to try people to moving forward? So one of the questions is like, how high can you go? I mean, you can produce in a plant, but there may be a limitation that you can generate, how much resistant starch. And the other thing is, if you generate, very high resistant starch, would it affect your physiology? Knowing the effect that Dr. Smith have talked about. So the other question is, how about comparing the whole food versus ingredient? Whether there are any difference, and so on and so forth. How about other food? For example, my favorite fruit, banana. It's resistant. It contains resistant starch, and it's RS2, and rice is R3. Is there any difference? The other thing is more related to analytical, is that whether there's a structure function relationship in terms of efficacy, as well as changes in the gut microbiome. And this is going back to Dr. Smith's work, is that if there are any protective effect, what enteropathogen, if you consume a food, the whole food versus an ingredient and things like that. And we really need to validate a lot of these role or important changes in gut microbiome, because we see changes, but is that changes really from the gut microbiome? And all these research, I think, need to take it to the human study level, because well, after all, these are my study, and then we want to validate a human. So at the end, maybe a bottle of Lafitte or Mouton work very well with the high resistant starch, and make you so happy and healthy. So with that, I'd like to acknowledge a bunch of all these people that from USDA that work with us, as well as University of Maryland, and a couple of visiting scientists from China. Thank you.
Great, thank you so much, Dr. Wang. And because we are bumping up on the end of time, we're going to just wrap up today's webinar. Although we do have a few interesting questions, we hope this was informative. And what we want to do is remind everyone that we will provide a link to the certificate after the webinar today, so that will come shortly. And we want to emphasize to everyone that IAFNS is holding its second annual Science Innovation Showcase next month, and the abstracts for that are due November 14th. If you go to our website under upcoming events, you can find all the information about how to submit an abstract. So if you got some interesting science going on, we would love to hear about it. And then finally, we want to thank all of our fantastic presenters today from USDA. We hope you learned a lot about the important work going on there. And if you'd like to connect with IAFNS and learn about our science that matters, this is how you can do that. And with that, again, thanks everybody, and we hope you have a great afternoon.